All right. So in this review, I've got a couple topics for everyone. We're going to go over some basic histology concepts, things that commonly confuse students. And let me pull up my document here. So these are just things in my experience that tend to trip students up. I'm also going to go over resources for you guys, and then we're going to put it together, look at some skin histology and bone histology, since that is the majority of what we've covered in terms of organ systems. And then I will also have some study tips for you, as well as 9 practice questions. Those will be histology focused, and I'm going to time them for you guys so you have an idea of how long you will have to answer 1 or 2 questions per station on the lab exam. So first off, you should know where your resources are by now. Everything is going to be in this resources module. And today I'm going to draw heavily from histology resources. And this is where we have our 2 resources, the Yale School of Medicine and then the Protein Atlas Dictionary. Today, I'm going to mainly use the Yale School of Medicine, and I think this is your strongest resource when you're trying to study from a microscope outside of class. So in here, we have covered epithelium, we've covered connective tissue, we have looked at the skin as well as bone. And histology at Yale has all of these labs. And just to recap, if you are wanting to study epithelium, for example, you're going to come in here, there'll be a bit of text at the top. So I want you to scroll past that. If you are looking for pictures, then those are going to be found in slides. So you could open up any slide here, and you're not going to be able to move it like you would on a microscope, but you will be able to see labeled features. And if you are wanting to quiz yourself, which I do recommend, you can hide those labels. So that's one way to use it. The other way, that's more like a microscope, would be to use any of these virtual microscope slides. For example, we talked a lot about the kidneys and how we find a lot of cuboidal epithelium as well as squamous epithelium in the kidneys. So if you were wanting to mimic what you do under a microscope, you're going to slap that slide under a microscope, zoom in from 4x to, this is our view more like 10x. Then from here, we can hone in on any of these small tubes. We'll learn later in the semester that they're called tubules, tiny tubes. So you're just basically looking for an opening, something with a little space in the middle, that lumen, and then we can start identifying this epithelium. In this case, we're looking at cuboidal, simple cuboidal epithelium. The way you can verify that is the cells are as roughly wide as they are tall. And then these purple dots are those nuclei. So you want to kind of hone in on this nuclei to find these epithelial cells. But that is just another way to use this resource when you don't have a microscope in front of you. So to my first point, let's talk histology. So this was our handy dandy chart for connective tissue. We don't have a chart for epithelium, but as I talked over in class, you need to know simple versus stratified epithelium, and you need to know your shapes really well. So some of those prominent examples are just like what we see on the Yale site. We talked about examples in the kidneys. We'll have a special case in the trachea, the skin, the bladder, as well as blood vessels. So I'm not going to rehash everything from the PowerPoint, but I just want to use slide examples to point out some of these different tissues. So we're actually going to pop back over to the kidney for a second. So if you recall, epithelium are going to be this layer or layers of cells lining an opening. And that opening could be the environment, like the first layer of our skin is facing the environment, or it could be more commonly a lumen, this inner opening of a tube. In the case of the kidneys, that is what we're dealing with. They have tons and tons of tubes here. So what we have, for example, this is a really good specimen, a lumen. So this is the tube itself, lined by epithelium, and then that white is like space inside the tube. So this is a picture-perfect example of cuboidal epithelium. In this case, we only have a single layer ringing that lumen, so it would be simple cuboidal epithelium. And to confirm, let's find a nucleus. So each of these cells has one nucleus, so we can see we have a single layer of cells here, And then looking at the shape, they are roughly as wide as they are tall. So that's one of the easiest examples to identify. It's a little trickier to find simple squamous. So let me just poke around here. If I can't find one here, we can always pop over to um, our static images. I'm gonna pop over here for a second. Try to find a really nice one for you guys. Okay. So we have moved on from the kidneys. This is a blood vessel slide. So we'll talk about blood vessels later. For now, all you need to know is that we have a layer of epithelium ringing the opening of the blood vessel. And just to set the scene for you guys, these are layers of connective tissue forming the walls of the blood vessel. So this part in the middle is the part which would be the inner passage containing red blood cells or erythrocytes. So we're going to ignore that middle part for now. But if we zoom in on this layer here, it's going to have a single ring of simple squamous epithelium. So this one can be a little tricky to find under a slide because it's such a thin layer. We're taking a single layer of cells and really stretching them out so they have that flattened appearance. And this is like what I was trying to find in the kidneys. So ringing blood vessels, we would see that single layer relationship. Let's see if we can just pull up one of these examples to really drive it home. Okay, make this go away. All right, so this is a nice slide. This is what I'm talking about. So we have this very thin flattened layer of cells forming that squamous epithelium. And in this case, because we just have a single layer, that would be our simple squamous epithelium. The other major type we would see would be our columnar epithelium. So we'll see that a lot of the gut, the GI tract, is going to be lined by columnar epithelium. And then there's also a special case in the respiratory tract. So we'll talk about both of those because both are fair game for the lab exam. Let's go down here. We're going to look at the intestine. I'm just trying to use more of these real life examples so that we're not just looking at static images, but instead simulating how it would be under a microscope. Okay, so what we have here is just a slice of the intestine. We're not worried about which region it is right now, but what we would have on that inner side is we have a lot of these, they're called invaginations. They're like inlets of the tissue, creating these villi. And so if we zoom in on one of those regions, what we have are these really tall cells. So they are column shaped, they are taller than they are wide. And again, if you're not quite sure, try to find the nucleus of one cell and then figure out that apical side where we have the part of the cell facing the opening. That's our apical surface. And then follow it back to this deeper connective tissue. So this would be that basal surface. So all these cells, just to recap, are sitting on a basement membrane. So based on that, we can figure out from the shape and from the layering here that this is a simple layer of columnar epithelium. Now let's build on that because we have a special example of columnar epithelium. There we go. And that is going to be found in our respiratory tract. So if we open up this trachea slide, and you could take this from any part of the respiratory tract, as far down as the bronchi where they enter into the lungs, but in this case, we just have a slice of the trachea. This is going to have a very special type of epithelium. So if we zoom in, just to give you some context here, this is going to be the part of the trachea lining the opening where air is passing through. So let's zoom in on that layer, let it load. Okay, so we can start to make out all these nuclei, and they are again anchored to the deeper connective tissue by that basement membrane. So let's get as close as we can. This would be like our 40x magnification. Now we can see that we have these uneven stacks of nuclei, creating this pseudostratified appearance. If we were to follow each of these cells down, we would see that each of the cells has an anchor to that basement membrane. So it's still a one-to-one relationship. We have a cell anchored to the basement membrane. Whereas if we were dealing with a true stratified tissue, we'd have these multiple layers of cell, and only that base layer would be attached to the basement membrane. So again, this is an apical side. This is where we have the cilia in this case, and then this would be that basement membrane. So this is that basal surface. So altogether, this would be called pseudostratified ciliated columnar epithelium. And because it's such a distinct tissue, it's a really good one to ask on the lab exam. Okay, so I'm not going to run through every single shape of epithelium in its stratified form. Like I mentioned in lab, stratified columnar epithelium and stratified cuboidal epithelium are a little rare, so they can be trickier to find. And they're basically just bigger versions of the simple epithelium. So, for example, the simple cuboidal epithelium makes up a lot of glands and ducts, as we saw in the kidneys. So the stratified version would just be a bigger gland. If we're looking at sweat glands, for example, they have a lot of stratified epithelium. They're just larger versions of the stuff that simple cuboidal would create. So there are some important examples, though. One of the biggest examples we talked about was the skin. So let's pull that up now. And we'll come back to this after we review our epithelium. So if you remember from when I went through this in class, this is our slice of skin. It is upside down in this view. So to orient you guys, this would be that apical surface facing the environment. Deeper, we would have our basement membrane. So this is the line or the border separating our cellular stuff from the deeper connective tissue. In this case, we have layers upon layers upon layers of stratified squamous epithelium. So that's one of the best examples of stratified epithelium that we have talked about so far. Um, as we talked about, the layers of the epidermis will start out more cube shaped. These actively growing layers towards the basement membrane are going to look a little more like the general cell model that we learned in our very second lab. But then as these cells get closer and closer to the surface of the epidermis, they'll start to flatten out and take on that stratified squamous shape. And then in this case, this would be thick skin. And so you have a really, really thick layer of that stratified squamous epithelium. Okay, moving on. That covers pretty much all the tricky areas for epithelium. We come back to our document here. This is our schematic for connective tissue. So if you're trying to study this, I would recommend going through the PowerPoint again, just running through all of those images, those examples that I gave you in class, because I tend to draw images or microscope slides that look like those examples. Depending on how you stain the tissue, connective tissue can look different under a microscope, but I'll try to stay true to what we looked at in class and what you looked at for your worksheets. Common struggle areas for people. Usually students are pretty good about telling their cartilage apart, so I'm not going to spend a lot of time on that. But it gets really tricky when we are looking at fibrous connective tissue because we have 6 different types. And if you were in class, you know that I ran through these and told you the ones we're most likely to see on the lab exam. But just to recap, loose areolar connective tissue is a really good one to ask. So is adipose tissue. We don't have a great example of reticular connective tissue, so that's less likely to appear on there. But you definitely want to know your dense connective tissue, dense regular, dense irregular, and elastic connective tissue. And those are what I'm going to focus on in this review. So let's jump back to Yale, and this time we're going to be dipping into the connective tissue lab. Okay, so we're just scrolling down. And again, this is a really good way to study is take one of these slides, delabel it, and quiz yourself or quiz a lab partner. Just practice that recall when you don't have the answer key in front of you. Okay, so let's see how this slide looks. Okay, it's the same skin slide. Cool. We'll come back to that one then. Okay. How do we want to do this? So I'll just go through the static examples then. So we'll start with our loose connective tissue and then I'll move on to dense connective tissue. Okay. So this again looks a little different than what we saw in class, but I just bring this up because um, you may see different stain colors. So your tissue may look more purple versus more pink on a side. But in this case, we're dealing with a loose connective tissue. So there's going to be a lot of empty space here, a lot of white shining through, a lot of light coming through from the microscope. So that's what gives it this much brighter appearance. And then what we have is a sort of an assortment of fibers. We have these thicker collagen fibers running, kind of crisscrossing the side. And then we'll have thinner fibers. So in addition to collagen, we talked about elastic fibers, the really curly fibers. And we also talked about reticular fibers. So to give you an example of how it might show up on the lab exam, let's find our connective tissue was all the way at the bottom. So I like to use this slide because it shows all three of those tissues. We had the really thick, lighter pink collagen fibers. We would find that in loose areolar connective tissue. Here we see those really squiggly, coiled, elastic fibers, and their properties allow expansion and recoil. So that's why they look a little bunched up on the side. And then the thinner hair-like fibers are those reticular fibers. So we would expect to see all of those on a loose areolar connective tissue slide. And this is the kind of example I would try to find for you under a microscope. I think the Yale one's a little weird, but it's worth it to point out that you might not always see the same color stain. It might be more purple, might be more pink. Okay, so that was loose areolar connective tissue. And yes, you can abbreviate that. If you give me LACT, I will know what you mean. Okay. So next up is adipose tissue and then reticular connective tissue. Okay, this one we can use the skin as an example. So you will find adipose tissue everywhere in the body. It provides padding, it provides insulation and long-term energy storage. So that's going to cover most of the body. But some places we can see it that we've already talked about would be in the skin. So just to reorient you guys, again, this is at epidermis, made of epithelial cells. Immediately deep to that, we would have that first papillary layer of connective tissue, and that is, yes, that is loose areolar connective tissue. Then a little deeper, we get into our dense irregular connective tissue. But beneath all of those layers, beneath the epidermis and the dermis, we have the hypodermis. And this will have many names. I think Dr. Yard might mention Camper's fascia in lecture, subcutaneous fascia. I tend to use hypodermis, but what we're looking at is mostly fat cells. All of these white puffy cells are those fat cells, and you can confirm if you're lucky, you'll get a nice cell like this, an adipocyte, where the nucleus is kind of shoved to the corner of the cell, and then the part in the middle is dominated by this vacuole containing energy storage, lipids, basically. So they have a really distinct appearance. They look like white puffy clouds on a cell. So that's a pretty easy one to ask of you on a lab exam. Okay. And then the last type, again, that was pop back to my list here. This particular one is tricky because we don't have a great slide showing that. So you don't really have to worry about it for the lab exam. Alrighty. And I did have a question about this from a student, so I do want to clarify, where would we find this reticular connective tissue? Really what happens is these reticular fibers, the stuff that looks like threads that you see in blue, those would weave between the cells of an organ system. So if we were looking at the kidneys, we might see these reticular fibers weaving through the actual cells of the kidneys. If we are looking at the liver, same deal. We have all of these cells of the liver, which are performing the actual function of the organ system, and they are supported by this scaffolding, this reticular connective tissue. That's why I don't have an example that's just like just reticular connective tissue. But we'll encounter it in organ systems as we study later in the semester. Okay. And then the other area I want to tackle today would be dense connective tissue. So this was the first one we started with. We talked about the difference between regular and irregular connective tissue. And then we also looked at our elastic connective tissue. So for this, I am going to use the static slides. So we'll start with the difference between regular and irregular. So the first thing I want to point out, looking at both of these slides, there's not a lot of light shining through on the sides. There's not a lot of white showing. So this is a pretty dense tissue. That's the first word we need. Is it dense or is it loose? And in this case, it's covering the whole side. There's not a lot of opening, so it's going to be our dense connective tissue. And then we need to figure out if it has a pattern. So in this case, we have all of these collagen fibers running lengthwise across the slide. And this is very similar to the one I showed you in the PowerPoint. So these fibers are forming a pattern. They're forming this kind of wavy pattern that we see here. Because of that, we can identify this as regular connective tissue. It has a regular pattern of collagen. So altogether, to get full points, you would need to give me the name dense regular connective tissue. Now, if we flip over to our other side, it's a little trickier to see. We have some collagen fibers running this way lengthwise, but then we also have like what looks like cross sections of collagen fibers, and then we even have some that are like creating these swirls. So based on this, we can't say if it's running in a particular direction. We can't find a pattern in the tissue. So that would be an example of dense irregular connective tissue, or DICT. So make sure you're giving me that full name, whether you spell it out completely or just give me the abbreviation DICT. Either is fine as long as you are giving me all of those details. It has to be dense and irregular. It's not enough to say dense connective tissue because we have two types. And then the last type that gets lumped under dense would be our elastic connective tissue. And in this case, I can show you this on an artery. So what we will be looking for is these elastic fibers. We'll be looking for these darker squiggly lines in the tissue. And in this case, we will find those lining the aorta. So I'm going to pop over to our blood vessels lab. And we're going to find a nice big artery to look at. See what this guy looks like. We'll try to find one with this different staining. Okay, so here's an example. The very innermost layer of the blood vessel will have a really thin layer of elastic connective tissue. So we have just this really thin layer of squiggly lines, and we also see them incorporated deeper in the artery. So they're a little faint on the side here, but we can see that we have these squiggles forming that elastic connective tissue. So if I'm trying to ask you this on the lab exam, I will be clear in the wording of the problem if I'm asking about epithelium or elastic tissue or other types of connective tissue, just because we just talked about how there are squamous cells lining the lumen of a blood vessel, but then we also are dealing with some elastic connective tissue, so that can be a little tricky. So if I were to ask you a question, I'd specify give me some connective tissue or give me some epithelium. I won't leave you hanging there. All right, let me just pop around, see if there are any last ones that we can see. Some elastic connective tissue. Okay, not so much here. So we have a slide in class that you may have looked at when you were examining elastic connective tissue that shows in a little more detail um, like this. So in this case, we're still dealing with a really pretty big artery, but you can see that the elastic connective tissue is a darker stain, and we can see a lot more of that darker elastic tissue on this slide. So I'll try to find an example where it's a little more obvious, but in any case, you'll want to look for these really squiggly elastic fibers to identify this as elastic connective tissue. Okay. So that covers kind of the common pitfalls for histology. And we're going to look at a little bit of skin histology now, as well as bone, and try to pull it all together. The trick will be finding a nice skin slide that shows things like our sweat glands and our hair. I do want to go into that a little bit with you guys. So if we go into the skin lab and scroll down, we're going to find a slide here called hair follicles. So this is what we want. This is going to give us thin skin that has hair on it. And again, it is upside down, so I apologize for that. But we'll just orient ourselves. So again, this would be the apical surface, the external facing surface. And then we have from this perspective, the epidermis looks pretty thin. And then all this swirly stuff would be our deeper dermis, our connective tissue made of that upper layer of loose areolar connective tissue, and then that deeper layer, which is that dense irregular connective tissue. And then even deeper, we would see that hypodermis, which is just full of these fat cells, these adipocytes. Okay, so we are now going to build on that. We've covered the connective tissue and cells that we'd see here, but let's talk a little more about features I could ask you under a microscope. So this will be specific to the skin system. So this is a nice example of a hair follicle. Remember, the part sticking up from the surface is the shaft. That's like the part that we can see. And then we have the root, which is the part that is embedded. And then we would have the bulb at the very bottom. And the bulb is that actively growing part that has a blood supply and a nerve supply. So it hurts if we pull hair out by the roots. You can get a little bleeding, and that pain is actually coming from the bulb here. So this is that active portion. One important thing to know is that this hair is contained in what is called a follicle. So that capsule is the follicle, and that is actually an extension of the epidermis. So if we zoomed in here, we'd see that this tissue is still cellular. So it's basically just a folding in of the epidermis surrounding the hair follicle, and then we also get this cuboidal epithelium forming that sweat gland. So that's what we're dealing with here. That's why it has this kind of dark outline around it, making up the follicle. So let's talk about sweat glands. We talked about three types: apocrine, merocrine, and sebaceous. The one that I'm most likely to ask you on a slide is sebaceous because it's the one that is really easy to find. So if we recap, the two types of sweat glands associated with hair follicles would be our sebaceous glands and our apocrine glands. The merocrine glands with an M, those are the free-floating ones. So they're not associated with hair. And then if we're looking underside, the only one that I will expect you to find is sebaceous. So this is the one that looks like kind of like a bunch of grapes bracketing either side of that hair follicle. And it's important to know that sometimes you won't have hair on your slide. Sometimes you'll just have maybe a hair that isn't completely grown out or a hair that is still developing, or you may even get an ingrown hair. But it will always have a follicle and will always have a sebaceous gland bracketing that follicle. So even when the hair is embedded deeply like this, still have a follicle, still have a little bit of hair that's growing out, and that sebaceous gland. So don't let that trip you up. Just because we don't have the hair, the shaft sticking out on the surface doesn't mean that we don't have a follicle or a sebaceous gland. So that's the main thing for skin. And then last thing I want to cover from an organ system perspective would be bone. Close some tabs here. Alrighty, so last thing we're going to pop into the bone lab. And same deal. We'll try to find stuff under a virtual microscope first, and then we'll look at some endochondral ossification. So to start with compact bone, this one looks very distinct. So even without zooming in, you can almost tell this is going to be compact bone because it typically is this black and white color on a slide. And then right away, we can start to pick out these tree ring shapes, these osteons, which are the basic unit of compact bone. That is something that is unique to compact bone. So do not Do not try to tell me this is spongy bone on the lab exam. This has to be compact bone. So if we were to zoom in on one of those osteons, let's say I like this one because it's pretty big. At the center of every osteon, we have a central canal or Herversian canal. Again, you can give me either name on the lab exam. And it's a little trickier to see on the slide, but what we have is these concentric rings, which are called lamella. A lamina is like a sheet or a layer. So in this case, we just have these concentric layers forming lamina, which make up that osteon. One thing that has been confusing students recently is figuring out canaliculi versus lamina. So the lamina is that ring. The osteon is made of lamina. I want you to kind of put that in one category in your mind. And then canaliculi are going to be completely separate. So the central canal here, let's start with that. This is that main passageway for blood vessels and nerves to supply the bone tissue itself, those osteocytes. So that's like our main interstate. But then this canal is not going to be able to reach a cell that is all the way on one of those outer lamella. Um, Instead, we have these little offshoots. These would be like exit ramps from the central canal. So we have these really tiny thread-like lines here representing canaliculi. So think of the canaliculi as these smaller off-roads that are going to these osteocytes. So they're always going to be radiating outward from that central canal. So completely separate from this idea of our lamellae. And the last thing that we can see here, you want to remember that the depression that we see in the bone, that little hollow, is called a lacuna. So we would expect that these lacuna are filled with cells, with osteocytes. I will not ask you I will not ask you to identify a lacuna versus an osteocyte on a slide. It's fair game on our model, but for here, it's enough to say that these are lacuna because it's a little harder to tell if they actually have cells in them. So just to recap, we have the central osteon unit, we have those concentric rings, which are lamella. We have the central canal, which is that main supply system, and then we have these much smaller canaliculi, these thread-like structures supplying the lacuna, which are all these little dark spots that you see. So that's our compact bone. Now, we also have spongy bone. Let's see. I don't think there's going to be a slide for this one, so we can just look at it from a static view. Okay, I borrow heavily from this website, so this is the same slide that you see in the PowerPoint. And this is what spongy bone will look like under a microscope. These slides tend to be pretty consistent. So what we have here is this pink connective tissue. And what you need to remember is that bone is still connective tissue. So it's mostly going to be collagen. We also have that hydroxyapatite component, that calcium rock-like component. But altogether, this does not make a highly cellular tissue. Instead, we're dealing with mostly connective tissue embedded with those lacuna housing cells. So don't get that twisted. A lot of people want to say that this part with all the cells in it is bone, but that's not correct because bone is mostly this non-cellular connective tissue. So the stuff in pink is the actual spongy bone. And as we learned, these little spurs or these little beams of bone are called trabeculae. So when these are all crisscrossed and knit together, they are what creates that spongy texture of the bone. Everything else you see on this slide, the stuff in dark purple and dark blue, those are blood cells. So this part surrounding the spongy bone is, in this case, bone marrow. So in this, we'd see blood cells. We'd also expect to see adipose or adipocytes, those fat cells. So there's a lot of stuff going on in here, but don't let that throw you off. This is bone marrow. This stuff in pink is the actual spongy bone. And that's pretty much the extent of what I would ask you from a microscope slide. All right, last thing before we get to the practice questions, I want to go over endochondral ossification. So, you think? Okay, we do have a microscope slide for this one. Good. Okay, so I had you guys spend a lot of time on this on the worksheet because it's such an important slide to know. This is how we get new bone. So if you get a fracture, we have to have some way to create new bone to replace that break. And this is that same process, when you're growing, your bones have to get longer. So we have cartilage, we have to have a process to translate that into bone. And that is what we see on the slide here. So you can definitely expect to see this on the lab exam. Okay, so you're in your 4x magnification. It might look something like this, maybe a little further out. So we need to get pretty close, maybe 10x or 4x magnification to start to see these different zones of ossification. So to orient you, the stuff on the left, this is that spongy bone that we just saw. So it's stained a little differently here, but we would have, let me get a little closer here. So what we have is this cartilage turning into spongy bone. And then the stuff in red would be some of those things like osteoblasts and osteoclasts that we talked about. So I'm not going to ask you to know that for the exam, but we see that we have this texture of interweaving cells. So figure out where your spongy bone is, and then let's confirm that we are looking at hyaline cartilage. So the stuff on the right would be just our default hyaline cartilage. And we do see that expected pattern where we have these pairs of lacuna. In this case, they house cells called chondrocytes because we're dealing with cartilage now, so that the prefix changes from osteo to chondro. So we've got chondrocytes just chilling in these lacuna. And then we have everything in the middle. So this can be a hot spot for students. If you don't know your zones really well, you will be kind of lost on this problem. So I'm going to get as close as possible. So first off, we have just our resting zone. This would be that default hyaline cartilage. It's not really doing anything other than its normal function. But once it gets a signal for new bone growth, we will see that it starts to rapidly divide. So we start to see these cells like stacking one on top of another because they're dividing so quickly. So that is where we get these columns of chondrocytes. And this would be that zone of proliferation because the cells are rapidly proliferating or rapidly dividing. Then they are going to undergo hypertrophy. So hypertrophy is where the cells are getting larger in size. We usually refer to it in terms of like muscle hypertrophy. Muscle hypertrophy is generally a good thing because you'll look more toned. You have bigger muscle cells. Same idea here. These chondrocytes are getting larger. They are becoming hypertrophic. So that would be our zone of hypertrophy, where we start to see these cells swelling up. They have a lighter appearance as well. And then we have a small zone of calcification. This is like a transition zone. So we're going from these hypertrophic chondrocytes, and now we are filling in with bone matrix. So those osteoblasts that we saw, they're going to come in and start laying down this bone matrix and filling in around those cells. So that would be this zone of calcification where we start to see a color change and a texture change. We're going from cartilage to spongy bone now. So it's pretty thin strip here. And then on the very left, we would have that fully ossified bone. So at this point, we're dealing with spongy bone. So your big tip for the lab exam is to focus on these middle transitional zones. Things like the resting zone, the zone of hypertrophy, the zone of calcification. Those are going to be really good test questions because that requires you to understand what hyaline cartilage looks like, as well as spongy bone. And it requires understanding of that process, how we go from just regular cartilage all the way to spongy bone. Alrighty. So in terms of study tips, I'll keep it pretty brief. I would do what I've been doing this whole time in this review is using those histology resources, especially the Yale School of Medicine site, going through all these labs and trying to look as you would under a microscope because those are not nicely labeled. It's very easy to go through this histology PowerPoint and say, okay, I can recognize that that is elastic connective tissue. Or, let's say, yep, that looks like pseudostratified columnar epithelium to me. It's easy when you have the answer in front of you. What's a lot harder is if we were to just take this slide, you know, I put an arrow on this region and I ask you identify this type of epithelium commonly found in the respiratory tract. Well, now you need to remember a lot of things. You need to remember pseudostratified, you need to remember ciliated, and you need to remember epithelium. So in 75 seconds, that can be a lot of pressure to recall an answer. So what we're practicing is that recall, taking an unlabeled slide, just like this example here. If I put the arrow on that layer, then you need to be able to tell me that's, simple cuboidal epithelium. So it's not so simple as recognition, the main challenge for you guys. I would also draw. So I think drawing helps a lot in lab because it is such a visual medium. It's not like lecture where you have multiple choice questions without pictures. This is all pictures or all models or microscopes. So the stuff that we do in lab with the worksheets, it's not just to fill time. If you're sketching out, for example, this endochondral ossification, that's going to help you to actually learn the patterns that we see on the tissue. So those are just two things to try. Those are things that helped me when I took the class, but challenge yourself. The goal of studying is to figure out what you know and figure out what you don't know at all. And that cannot always be a comfortable process. Sometimes it requires, maybe going through these practice questions I'm about to show you, and you try to answer them, maybe you don't know anything. That can be valuable too. That's your wake-up call that you should start reviewing these things, and just keep practicing that recall. It's better to do that now and to mess up now than when you have a blank lab exam sheet in front of you and you don't recall anything. So that's just my 2 cents. But let's go ahead and segue into those review questions. So that is how I will end this review, and I will be timing it. Just use my phone here. And we're going to go through it just like we would at the start of the lab exam. So we'll just present this. Maybe. Okay. So typically on a lab exam, we'll start with 8 PowerPoint questions. We actually have 9 here. I wanted to add one that will be showing up on the lab exam for sure. Let me pull up my timer here. But every lab exam, we will start with those first PowerPoint questions, and you will not be moving for that for those first 8. Just to ease some anxiety, answer some questions as well. You won't have your stuff with you, so we'll put that on a side counter or at the very back wall of the lab. Of course, you do need to be silent. You need to store your devices. We will do a device check, so I need to make sure you don't have a smartwatch, phone, AirPods, anything. Those all need to be away. All you will need is a writing utensil. And then you'll grab an answer sheet from me. You'll grab your question sheet. I'll take my attendance via that answer sheet, so you won't have to worry about signing out or anything. And then you'll find your assigned station. So I'll have, let's say, station 18 circled on your sheet. There'll be numbered stations where you normally sit in those 5 rows, and you'll just find your seat, and that will be your starting point when we start rotating through the questions. No talking. Eyes on your own paper. We do take academic dishonesty very seriously. And I have had incidents in the past where, we had to have a hearing with students. It can get pretty stressful. Dr. Yard gets involved, and it can even be elevated to dean of the school, the university level. So we don't want that. The only guaranteed way to get a 0 on the exam is by cheating. I've never had a student get a 0 that didn't cheat. So again, I'll get off my soapbox, but do make sure that you are submitting your own work and not someone else's. But regardless, we will rotate every 75 seconds. So as we're going through these slides, I will tell you when you have 15 seconds remaining, and then I'll say switch, which is what we would do normally. We'd get up, move to the next station, and then sit and answer those questions. A couple things to know are I do not want you to touch the models. Those are set up specifically if students are moving them around, arrows can move around, they can get a little messed up. So we'll keep hands off the models. If you need to adjust a microscope, you can use that fine adjustment, but otherwise, do not mess with them. And then at the end of the exam, everyone will have 10 minutes to review any station or PowerPoint slide. So you won't have time during the exam to go back, but at the end, that is your time to review anything you weren't sure about. Okay, so here we go. We'll start timing now. 15 seconds remaining. Question number two. Fifteen seconds. And switch. Question three. Fifteen seconds. And switch. Question four. 15 seconds. And then switch. Question 5. Fifteen seconds. Switch. 15 seconds. Question 7. Fifteen seconds. Question 8. 15 seconds. And last question. 15 seconds. And time. So that concludes our practice questions. I'll run through the answers with you real quick, but hopefully from these sample questions, you get the sense that I do give hints. So while you're not required to know that our chondrocytes rapidly replicate during that zone of proliferation, if you do know that, you can almost answer this question without seeing the slide. So this can be very helpful hints. So I would, again, go through the PowerPoints, look at the physiology, the very brief physiology that I talk about in relation with all these tissues, and that may help you out on the lab exam. But let's go through these answers. We'll just start at the back, make our way back to the top. So this is asking us to identify the specific zone of endochondral ossification. In this case, the chondrocytes are rapidly dividing, creating these columns of cells. So this would be our zone of proliferation. We just looked at this tissue as well. So it's asking us to identify this tissue, which is formed out of these little beams. So that is our clue that we are talking about spongy bone. 7 asks the tissue, well, we'll have to start with 6. So name this layer found only in the thick skin of the palms and soles of the feet. This is a huge hint. So if you already know your layers and you know which one is present in the thick skin, you'll know that that is stratum lucidum. But we can also confirm this layer is slightly lighter on the sides. It's allowing a little more light through in the microscope. So that is that stratum lucidum. And then the follow-up here is question 7. This tissue can be classified as blank blank epithelium. So this is going to require you to understand that we have multiple layers of stratified epithelium. So in this case, it would be stratified squamous epithelium. Okay. This is involved in long-term energy storage and insulation. So if you can't tell right off the bat, this is adipose tissue. We can confirm because we know adipose tissue is for long-term energy storage. It has all those lipids stored in that big vacuole. Number 4, this type of connective tissue is resistant to forces in many directions. So just looking at this, we can see that this is a dense connective tissue. It's covering the whole slide and there's not a lot of room for light to shine through. So that narrows our options to the dense options. And then if we look at it, we can't really distinguish a pattern here. There's tissues running all different ways. So we know it's dense irregular connective tissue or DICT. Number 3 asked, this type of cartilage is found between vertebrae and other areas of high wear and tear. So that's our big hint here. High wear and tear is referring to fibrocartilage. So to confirm, we know fibrocartilage has these columns of chondrocytes. So they look kind of like peas in a pod. They're interspersed between our collagen and the matrix of the cartilage, all that mixed together. So this would be our fibrocartilage. Question 2, found lying in the upper respiratory tract and trachea. This type of tissue is called, we just looked at this example, But let's examine that basal layer. We see uneven stacks of nuclei, okay. And we also see this cilia, this ciliated layer facing the apical surface. So based on those clues and based on the fact that it's in the respiratory tract, we can identify this as pseudostratified, ciliated columnar epithelium. So I do want you to give me that full name. It's not enough to say columnar. Try to give me that whole pseudostratified ciliated columnar epithelium. And then last question was the indicated tissue is found at the ends of long bones and is called what? So big hint here, at the ends of long bones, we would expect to find hyaline cartilage. To confirm, we look at this tissue, we see these pairs of lacuna housing, in this case, chondrocytes. So this is going to be hyaline cartilage. So hopefully that gives you an idea of how I'm walking through these questions when I write them and the process for answering them. I do like to give context clues, so the information from the PowerPoint can become these nice hints to confirm the tissue that you're looking at. So I hope this helps. You can review this as often as needed, ask questions about it in class if you have them, and happy studying.